Hello everyone, I am Nivedita Jaina from Ambika Prasad Research Foundation. Welcome to our future channel, Biodiversity and Conservation. So today we are going to discuss about the applications of bioinformatics in biotechnology. So let's start. So as we know that bioinformatics is related to genetics and genomics, it is a scientific subdiscipline that involves using computer technology to collect, store, analyze, and disseminate biological data and information, such as DNA and amino acid sequences or annotations about the sequences. So the bioinformatics tools aid in comparing, analyzing, and interpreting genetic and genomic data, and more generally in the understanding of evolutionary aspects of molecular biology. At a more integrative level, it helps analyze and catalog the biological pathways and networks that are an important part of systems biology. And bioinformatics has got so many applications in biotechnology field. Some of the important ones include automatic genome sequencing, automatic identification of genes, identification of functions of genes, predicting the 3D structure modeling, and pairwise comparison of genes. So, in automatic genome sequencing, bioinformatics are used in genome sequencing techniques such as bioinformatics are used in the development of automated sequencing techniques that use PCR, which is also known as polymerase chain reaction, or BAC, which is also known as bacterial artificial chromosome-based gene amplifications, two-dimensional electrophoresis, and also automated reading of nucleotides. These are used in joining the sequence of smaller fragments of DNA to form a complete genome sequence. These are also used in the prediction of promoter region of the genome and promoter coding region of the genome. Small fragments of genome can be amplified using polymerase chain reaction or bacterial artificial chromosome. Amplified fragments may suffer from nucleotide reading errors, repeats. These in turn generate multiple copies of the same genome fragments, and these repeats are removed just before the final assembly of all genome fragments by mathematical models. So then coming to the identification of genes, the first one is GRAIL, which is one of the most commonly computational tools used for ORF identification. This tool provides important information such as splice junctions, translation start points, and non-coding scores of 60 base regions on both sides of the putative exon. And the second one is Glimmer. Glimmer is a software used for finding genes in microbial DNA, especially the genomes of bacteria and fungi. And the gene locator and interpolated glimmer uses interpolated IMMs to recognize the coding regions and differentiate them from non-coding DNA. The third one is gene scan. This tool is used for the identification of complete gene structures in genomic DNA for various organisms. It can predict exotic gene structures of genes as well as locations in genomic sequences. Then coming to the identifying gene function. So after identifying the open reading frames present in the genome, next step is to annotate the structure and function of the genes. So bioinformatics tools such as sequence search and pairwise gene alignment technique are used to identify the gene function. The major four algorithms such as BLAST, BLOSSOM, Cluster X, and SMART are used for the functional annotation of genes. Then in three-dimensional structure modeling, a single protein may present in different conformational states depending upon its interaction with other proteins present in the vicinity. And three-dimensional structure of the protein has got some regions exposed for protein-protein or protein-DNA interactions. Also, the function of these proteins also depends upon these interactions. So bioinformatics techniques are also used to predict the possible conformation of the protein coded by a gene. This in turn helps in identifying the function of the same protein. In pairwise genome comparison, after the identification of functioning of a particular gene, bioinformatics is used for pairwise genome comparisons, and this pairwise comparison of a genome with itself is done to get the details of paralogous genes or duplicated genes that have the same base sequence with some functional variations. Also, this comparison done against other genome to get information such as orthologous genes. These genes are equivalent genes present in two different genomes due to speciation. And this technique is also used to identify different types of gene groups and adjacent genes that occur in the close proximity as they are involved in common higher level function and also lateral gene transfer, that is transfer of genes from a microorganism that is evolutionary distant. And bioinformatics technique is also used to analyze gene fusion or gene fission, gene group duplication, and much more. Bioinformatics techniques are also used in the process of drug discovery, and this speeds up the process and also used in the effective and efficient designing of vaccine and also in the designing of antimicrobial agents. So bioinformatics has got major impacts on biotechnology and its applications. The vast amount of data generated by human genome project or by other genome sequencing project would be unmanageable without the bioinformatics techniques, and without bioinformatics handling interpretation of this data would be unthinkable. And thank you.